I'd like to start by acknowledging that I'm joining you from Balladong Noongar country and pay my respects to the massive contribution that Aboriginal people make to NRM in the wheat belt of WA. We're really lucky to have several of our elders in the office today for their quarterly advisory committee meeting, and I pay my respects to them, to those who have gone before them, and those who are rising in our community. I'll just take a minute to introduce Kattie. Dr. Weigh is a postdoctoral research fellow for the Department of Primary Industries and Regional Development Soils Research Team based in Wollongbar in northern New South Wales, with a background in molecular microbial ecology, bioinformatics and biostatistics. She currently works on 3 projects, including investigating the power and potential of soil microbial indicators with the Soil CRC, where they're conducting controlled environment and isotopic enrichment experiments. They're developing next generation soil techniques to address multifaceted soil constraints with the GRDC Unlocking Soil Potential project and designing novel amendments to capture carbon for long-term stabilisation in soil with the Soil Science Challenge. Dr. Weigh's goal is to advance understanding of the mechanisms through which soil microbial communities drive ecosystem processes, support sustainable agriculture, and enhance climate resilience. Dr. Weigh will be presenting the results of a WA wheat belt-based project that was introduced at Talkin Soil Health in 2022. We were all set to hear all about the results of some of that work at Talkin Soil Health in 2025, but Cyclone Alfred had other ideas for Kattie. Unfortunately, she was unable to attend due to the impacts of the cyclone. So we're really happy, Kattie, that you're able to join us today and share that information. We've had a lot of requests from community to hear the results of that project. So I'm really excited to hear what you have to say. So without further ado, I'm going to pass over to you, Kattie.
Okay. Thanks very much, Timothy. I'll just organise sharing my screen. Okay. How does that look for everyone?
Yep, I've got your cover page for your preso showing nice and clearly. Thanks, Kattie.
Okay, great. Thank you very much. So quantifying soil biology, what, why, and how. So today I'll introduce the case study, the sites that we studied, and we'll explore some differences in microbial indicators, both between sites and within sites, using the WNRM as a site-specific example. So in this webinar, I'll be presenting findings from a project that was co-funded by the Soil CRC called Soil Microbial Indicators, What Do They Mean and How Can They Be Used, which is led by Mick Rose. So there's a huge interest among growers and the community to analyse soil microbial communities for the benefit of soil function and agricultural production. But there remains very little consensus on which variables to measure, how to interpret results, and how to integrate results into soil health assessments in a meaningful way. Making sense of microbial indicators and getting more knowledge around how these can be used and interpreted will improve on-farm decision-making, allow growers to track changes in soil health over time, and enable data-driven improvements in soil function, ecosystem service provision, and agricultural productivity. So soils are rich in microorganisms, harbouring the greatest diversity of any other ecosystem. Through chemical and biological mechanisms, soils drive important ecosystem processes that sustain life on Earth. These include the provision of clean air, water, provision of food, fibre and fuel, climate regulation, and opportunities for pharmaceutical discovery. Within an agricultural context, there's obviously a desire to monitor soil attributes and functions. So there's a range of tests that we can explore to characterise healthy, biologically active soils, including those that focus on carbon fractions, key populations such as pathogens, nematodes, and arbuscular mycorrhizal fungi, or AMF, soil microbial biomass, enzymes and functional activities. We can also look at agronomic outcomes like yield, plant biomass, and nutrient uptake that are influenced by these soil factors. So clearly there's a huge range of indicators that can be analysed, all with varying accessibility, costs, standardization, sensitivity, variability over time, and interpretability of the results. But throughout today's webinar, you'll see that soil protein is now more interpretable and sensitive to management and is a meaningful biological indicator that delivers soil health benefits. So in this project, we studied 5 sites across Australia, including the Wheatbelt NRM, Central West Farming Systems, Birchip Cropping Group, and Sardi in South Australia. We evaluated the effects of management practices on soil biological indicators. And with the help of these growers, 89 soil variables were surveyed, including nutrient and carbon fractions, microbial biomass, qPCR of nematodes, pathogens, AMF, enzyme activities, and more. So across 6 sampling times over pre-sow, in-crop, and post-harvest in 2022 and 2023. So here we'll focus on the 2022 data set. So the WNRM case study focused on 2 sets of paired sites, both with 1 conventionally managed site and 1 under regenerative practices. Both regen farms use zero-till, multi-species cover crops, and integrated livestock and intercropping rotations. One of the regen paddocks used organic inputs such as compost and fish extracts, while the other retained stubble, rotated monocultures, and used seed coatings as biostimulants. It's worth noting that the other study sites in this project focused on more contained experimental size plots that were within the same geographic location. But the WNRM paddocks each pair was in 2 different locations. All right, so if we look at fungal, nematode, and enzyme compositions, firstly, I will be presenting a few of these plots throughout the presentation. So the biggest thing to note is that samples that are positioned closer together within a plot share greater similarity, and samples positioned further away from each other share less similarity. And the colours in these plots represent the study site, and the shapes represent the sampling time, whether it's pre-sow, in-crop, or post-harvest. So here, the biggest differences observed were between sites, with comparatively more subtle differences observed by the sampling time. And we can see a bit of a spread of the sampling points for the WNRM site, particularly in the fungal and nematode composition plots. So here I've overlaid what's called a SIMPA analysis. It basically shows which are the biggest drivers of the differences observed between the fungal, nematode, and enzyme compositions. It's worth noting that we saw congruency between enzymes tested at native soil pH versus pH 7. This is probably because we're studying agricultural soils where a neutral pH is generally maintained. So let's take the pre-sow data set as an example, and we can explore the chemical soil variables that best explain these observed differences. So here I've used a bioenv algorithm to determine the best model that includes 3 or more explanatory variables for these differences. So we can see here that there's a mixture of controlling factors, indicators, and outcomes that indicate changes in fungal, nematode, and enzyme composition. There's a mix of different carbon and nitrogen sources, soil pH, as well as protein content. And again, we see here that the NRM samples are a little bit more variable and spread apart, which is likely attributed to larger scale confounding variables like soil attributes that are associated with those individual locations of the paired sites. Okay, so if we look at the distribution of the measured variables, these are just some of the variables measured at the WNRM site. Each column represents a different one of the paddocks that have then been organised into pre-sow, in-crop, and post-harvest sampling times. Each row represents a different measured variable. The dark shading indicates a higher abundance or a higher number of that specific variable. But basically, let's just jump into some of the key conclusions from what's been found here. So we can see in the top heat map, the microbial biomass heat map, we observed a greater amount of microbial biomass at the post-harvest sampling time. This is likely due to the affiliation that microbes form with plants in the rhizosphere, feeding on their photosynthetically fixed carbon and performing nutrient cycling functions for the plant potentially. We also observed high mineralizable nitrogen content. We observed an alignment of that with enzyme activity. We also observed a relationship between, or a consistency between soil protein and enzyme activities, showing similar patterns across samples. If we look particularly at chitinase, we can see a potential alignment or correlation there. So if we examine this a little bit closer, these are distributions of soil protein on the x-axis and chitinase activity on the y. And there's a plot for pre-sow sampling, in-crop and post-harvest sampling times. So important to know is that chitinases are involved in fungal degradation and plant defense. And ACE protein is an indicator of soil organic nitrogen. ACE protein reflects microbial investment in protein degradation, reflecting microbial functions to acquire nitrogen from organic substrates. So this is functionally linked to chitinases, which microbes use to break down chitin, whether it's in fungi, nematodes, insect residues, or root exudates. So together, these functions indicate microbial nitrogen acquisition. And beyond disease suppression, soil protein delivers further benefits for soil, which include soil aggregation, resulting in improved microbial habitat, aeration, water retention, nutrient cycling functions as well, as we looked at in the enzyme activity in the previous slide. So if we look more locally, did we see these patterns at WNRM? Yes, we saw strong trends between the soil ACE protein and chitinase activity on a more local scale using the WNRM as a case study. This was also consistent at all sampling times. We can see the top left is a plot of all of the sampling times together, and then I've broken that up into pre-sow, in-crop and post-harvest. So we know that soil protein is related to mineralizable nitrogen, both of which are important to soil microbial functions, as demonstrated through the enzyme activities. So this is some work by Mick Rose and others on study sites within the MAC research site. They demonstrated that measures of easily available labile carbon are not only correlated with each other, but they support microbial biomass, as shown in the left plot. We have microbial biomass on the y-axis and hot water extractable carbon, which is a labile source, on the x-axis as a predictor. We also saw that labile carbon supports enzymes involved in carbon, nitrogen, and phosphorus turnover. Results I haven't presented here show a similar but weaker trend for WNRM, but the weaker signal is likely due to the confounding factors I mentioned, like the different site locations, which have different soil attributes. And we actually did some DNA sequencing on the baseline pre-sow 2022 samples. So there's many different types of DNA sequencing approaches that can be taken to analyze soil microbial communities. Here we did what's called phylogenetic amplicon sequencing. Basically, DNA is extracted from a soil sample. The genomic regions unique to bacteria and fungi are selected and enriched by PCR, and then they're sequenced, which allows us to identify the members of the soil microbial community. So if we look at fungal and bacterial richness and diversity across all of the study sites, richness is basically the number of species in a sample, and diversity takes into account the number of species in a sample, as well as how evenly distributed those species are among groups. So basically, the higher number here, the more diverse. So we can see differences across sites, followed by more subtle differences within sites, which are likely attributed to management. I tested the diversity disease suppression hypothesis here. Basically, we have pathogen load on the y-axis of all of the plots. Bacterial richness and diversity is on the top row. Fungal richness and diversity is on the bottom row. So basically, we'd expect a negative trend, as we're seeing here, to support the hypothesis that diversity leads to disease suppression. So despite the small sample size, or relatively small sample size, of 5 sites sampled, we see some indication that this hypothesis is supported. An interesting next project or next step might be to measure a few things on a bunch of soils to see if we can get more clarity around these sorts of trends. And it would be interesting to see whether bacterial or fungal diversity is more important in pathogen suppression. So if we look at WNRM, bacterial and fungal richness and diversity, again, we have bacterial richness and diversity on the top row and fungal richness and diversity on the bottom row. So we can see that, we can see that conventional 2 and regenerative 2 from the same pair, they're actually over the fence. The regen paddock showed greater fungal richness, while the conventional paddock showed greater bacterial richness. This could be a result of management or of pH, or a combination of the two. So if we look at the typical distribution of bacterial richness versus fungal richness with pH as present on the x-axis, we can see that bacterial richness increases towards a neutral pH, but fungi peak at around 5.5 or 5 to 6. It's worth noting that bacterial and fungal biomass follow very similar trends to this, to these richness curves. Okay, and now if we look at the pH of each of these paddocks, the conventional site has an optimum pH for bacteria, and the regenerative paddock has an optimum pH for fungi. So this is likely a result of the difference in pH, which could be an outcome driven by management. Okay, if we look at microbial community composition across all sites, we can see some pretty distinct microbial community compositions for bacteria and fungi across the sites. Again, we do see larger differences within the WNRM paddocks. Okay, now if we look at the WNRM bacterial and fungal community compositions, we can see that the paddocks show different community compositions, but the paired sites share more similarity, likely due to their close geographic location. Now, this is quite a complex figure, so in the interest of time, I won't go into this in detail. Basically, this is showing the abundance of different microbial populations by paddock. So the key findings for the dominant taxa in the bacterial community were that Bacillales were enriched in conventional farm 1. Typical functions of this population are nutrient cycling and chitin degradation. So this enriched population may be associated with the higher enzyme activity and mineralizable nitrogen that we saw in conventional farm 1 earlier. Rhizobiales was enriched in conventional farm 2. Rhizobiales, as many of us may know, are associated with root health, forming root nodules on legume plants and aiding in nitrogen fixation. Rhizobiales are likely enriched in conventional 2 because that paddock was used to grow lupins. And if we look at the fungal heat map of dominant taxa, the key findings here were that Aspergillacea was enriched in conventional farm 1. Typical functions include organic matter decomposition as well as nutrient cycling, with some reports suggesting their involvement in phosphorus solubilization to produce plant available phosphorus. Another population, Pleosporaceae, was enriched in regen farm 1. It's a known plant growth promoting fungi that's been associated with disease suppression. So there's plenty of digging we can do in this sequencing data, plenty of more work to do. Plenty more work to do. So yeah, we'll be writing up papers and look forward to presenting more results on this sort of stuff at the next opportunity. So the next steps are sort of linking it all together. So building on how soil variables and climatic variables influence microbial community attributes, and then how those then influence soil function, pathogenicity, and plant health. It'll also be interesting to improve our knowledge of the resources that drive things like soil function and pathogenicity. Just as we saw with the labile carbon fractions, fueling microbial biomass and functions, such as those involved in carbon, nitrogen, and phosphorus turnover. And as we saw, the different chemical conditions that select for certain microbial populations. So there's a lot that can be measured in soil to give interesting and meaningful insights into what's happening. But something like DNA sequencing, for instance, is certainly not the most cost-effective or interpretable approach, hence why it's much more commonly used in research than adopted by growers at this point in time. So if we're wanting to prioritise, soil-based protein seems to be a good candidate for a soil health proxy, given its strong association with mineralizable nitrogen, aggregate stability, and enzyme activities. A focus on increasing soil labile carbon supports microbial biomass diversity and function. Greater microbial diversity is likely to result in disease suppression. So key things we want to manage to promote biologically active soils would be, to build soil carbon, which is fuel for microbial life, fuel for soil function, manage soil pH. While this study just showcased a relatively limited pH range, pH is a major regulator of microbial abundance, diversity, and function. And moisture content. Water drives everything: carbon and nutrient availability, decomposition, respiration, pH, and so on. So the biggest drivers that influence soil microbiology are obviously the soil factors that we've discussed, like soil moisture, pH, texture, carbon fractions, and so on. The larger scale climatic variables, as well as management. Thank you very much for your time and the opportunity to present these results. I'll open it up for any questions.